Hello, Dr. Cisse covering anatomy and physiology. Today we'll talk about chapter 4, transcription and translation. On your left, you see a tRNA, transfer RNA. On top, you see DNA molecule. When you go into the DNA to look into the detail of the DNA, you see nucleotides. You see the nucleotides here, 3 of them, triplets. We have a copy of this DNA called the mRNA in red. Then below, we have tRNA with amino acid attached to each one amino acid per tRNA. And then you see pieces of this amino acid coming together to form a polypeptide, which will be the protein. Now, the video will explain how proteins are made in the cell. In order for us to understand that, we're going to take an example of baking a cake. If you want to bake a cake, you need a recipe. Let's say your grandmother has a recipe book in the kitchen, and that book always stays in the kitchen. Then, in order for you to see the specific recipe of a cake you're trying to make, you have to flip it with your fingers, right? Flip the pages of the book. You flip it, you open it, and from the book, you make copies on a piece of paper. Now, what you write down on the piece of paper are the ingredients you need to put together to make the dough to bake the cake. Then, after you mix that, you shape it, you put it in the oven, you bake it to make the cake. Now, if you think about it, the cake came from the book, but the ingredient that made the cake came between the book and the cake by some ingredients you'll find in your kitchen, in the pantry, to be able to make the cake. So in this case, we have a book, we make a copy of the book, we pick up ingredients, add together based on the instruction on this copy, that is just a copy of the book, to make the cake. It is the same thing as proteins are made in our body. The book in this case is the DNA located in the nucleus. So in this case, the kitchen is the nucleus of the cell, just like the middle of the house, right? Then somebody it's an enzyme called polymerase that will write down the copy of this recipe on the piece of paper. Then, by using the ingredients, we bake the cake. So the copy of the DNA is called mRNA, messenger RNA. Then, the ingredients that need to be added together will be brought back by the tRNA, the amino acid, to make the protein. Because we said proteins are just pieces of amino acid added together. That's why we understand who builds those protein together. Now, the oven in this case is a machine called ribosome that put together the pieces of amino acid to make the protein, in this case, the cake. So, what is the connection between DNA and RNA? Now we know the RNA, for example, the mRNA, is just a copy of the DNA. But mRNA, messenger RNA, is not the only type of RNA. We got 3 types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. But what is RNA? Just like DNA, it is the nucleic acid. Remember, we have 4 types of macromolecule: carbohydrates, proteins, lipids, and nucleic acid. So RNA, just like DNA, is a nucleic acid. But we have 3 types of RNA. And then guess what? RNAs are smaller than DNA. And what are the differences? First, remember the DNA was a double helix? RNA is not double helix. It's just a one nucleotide chain. It's just a one chain. And then DNA, the D stands for what? Deoxyribose, which is a sugar. Now, we don't have deoxyribose in RNA. We have just a ribose, the sugar. Then in the nucleotide, remember in DNA we have A, T, C, G. Now the T is replaced by uracil. So instead of having T, we have U. And then if you take RNA, it is usually in the cytoplasm. But you know the DNA is in the nucleus because the book is in the kitchen is the nucleus. Now let's ask some questions now. What is the gene? We talked about it in the other video. The gene is the piece of the DNA that can code, right, for a protein. So that DNA has to make mRNA and then protein. We'll talk about genetic code. We'll talk about how DNA codes for protein structure, how amino acid assemble into protein. So now we know the building block of protein is amino acid, right? So amino acids come together to form protein. So and then we'll talk about what happens when you do it, when you make the protein, and what it means to express a gene, and how do you make stuff that are not proteins in the cell. So what is the gene? A segment of DNA that carries the code for a particular protein. It's a piece of DNA that can code for a protein. So an information containing segment of DNA, remember that's only 2% because 98% of the DNA is not coding for protein. Only 2% is. That codes for a production of a molecule of RNA, remember mRNA, that plays a role in the synthesis of one or more protein. Okay? So amino acids are the building blocks of protein. So the tRNA has to bring the amino acid to come together. What is the genome? All the DNA. We have 46 human chromosomes. And what is the genomics? A study of the whole genome. What is genetic code? Body can make millions of different proteins. We call all those proteins together the proteome. But guess what? We only have 20 amino acid coding for that. Just like we have 27 letters in the alphabet and we can write books, it's the same thing. It is just the way they come after each other. Their combination determine what it is. What is genetic code? A system that enables these 4 nucleotides to code for amino acid sequence of all proteins. So only 20 amino acid in 3 nucleotides per amino acid. Each amino acid is 3 nucleotides. We call them base triplet, right? Now, the base triplet is the sequence of 3 DNA nucleotides that stand for 1 amino acid, right? The codon is the 3 base sequence in the mRNA. So if you think about it, the base triplet is in DNA, and if you make the copy in the RNA, those 3 will be the codon, okay? And then the anticodon will be the one on the tRNA that has to bring the amino acid to build the protein. How does protein is synthesized in the body? So we have the same DNA, right, in all cells except for cell and some immune cells. But the same genes is not always activated in every cell at the same time. Every gene will be expressed in different cells depending on the situation. Now let's talk about the copying of the recipe. In our example is transcription. What does that mean? The book is the DNA. The magic finger polymerase flip the pages of the book, right, to open it and to build the mRNA. What is transcription? Transcription is the making of mRNA from DNA to make the copy of the recipe from the book. How do you bake the cake? The baking of the cake is called translation. From the copy of the book on a piece of paper, which is the mRNA, you took into your house to use the ingredients, or those ingredients are called nucleotides. Then, because the nucleotides are the pieces of the mRNA, then you put them in the oven. In this case, the oven is called the ribosomes inside the cytoplasm to make the cake, which is the protein. And then the tRNA are the one that bring the amino acid based on the code they read. And we call the Tiffany, the tRNA Tiffany, right? So translation is the making of protein from mRNA. So basically, from DNA, we make mRNA and then we make protein. When a gene is activated, its gene is expressed, messenger RNA, mRNA is made. So when the gene is activated, we make mRNA. That mRNA is just a copy of the DNA, the part of the DNA that's being expressed. Now we know what is the gene. It's just the part of the DNA that's being expressed. That's why it's called the complementary to the gene. Not to the whole DNA, just the gene, that little part. Now this mRNA has to migrate, has to move from the nucleus to the cytoplasm because it's smaller, where it codes for amino acid. So the process of protein synthesis starts with the DNA to mRNA to protein. The beginning is transcription when we made the mRNA from DNA in the nucleus. But in translation, which happens in the cytoplasm, from that same mRNA, we make proteins. So in translation, the mRNA codes for protein. It happens in the cytoplasm. So what is transcription? The beginning, DNA to mRNA. Copying genetic instruction from DNA to mRNA. What machine does it? What enzyme does it? The enzyme is called RNA polymerase. It is the enzyme that binds to DNA and assembles the mRNA. Certain DNA-based sequence signals start. It means when they see ta-ta-ta, it's called ta-ta box, the polymerase would start making the copy of the DNA to mRNA. Then where it finds the C on the DNA, it adds the G to the mRNA. So where this happens, in the nucleus, when it finds C on the DNA, it picks up a piece of G to start building the mRNA. So G it's at C, T it adds A, and A it adds U. You see, in this case, we don't have the T in the mRNA, so it's replaced by the U. So transcription makes mRNA from DNA, but by RNA polymerase, which rewinds open the DNA helix behind it. Now, gene can be transcribed by several polymerase molecules. Terminators are base sequence at the end of gene signaling stop. At the end of the gene, the machine will read the stop signal. Now, it makes the pre-mRNA. It makes like a rough mRNA, immature mRNA. And then it has to remove all the junks from it and then keep the specific part that will be coding from will code for the specific protein. Enzymes within the nucleus remove intron from the RNA and splice exon together. Now, the way to remember this, the exons are the one that are expressed. They're the one that we use. The introns are removed between them. So that's why we have alternative splicing. We have to cut the mRNA, the pre-mRNA. We have to remove all the intron to keep the exons to put them together in different combination to produce different types of proteins. Now you can see here that the adding up of different amino acids by the ribosomes will determine the making of the protein. But at this point, the mRNA to the protein is called translation, right? Transcription is the beginning process, which is the DNA to the mRNA. What about translation? From that same mRNA to make protein is called translation. Where does it happen? In the nucleus. Three main participants in translation. The mRNA itself that carries the code from the nucleus to the cytoplasm. This is like our recipe copy. Then a tRNA is called transfer RNA that has to deliver a single amino acid to the ribosome, which is the oven. So the amino acid here will be the ingredients to make the proteins. Then the ribosomes is the organelle, the machine that has to read the message, that has to translate It's like the oven has to change the dough into a cake. How does translation happen? Messenger RNA molecule begin with a leader sequence. So translation has to act as binding site for a small ribosomal unit, and then the ribosome has to be added. And the tRNA has to add the amino acid, small RNA molecule, coil on itself to form an angular L shape, right? The one that I showed you at the beginning. The tRNA picks the free amino acid in the cytosol, floating around, and add them together. This is the tRNA. It has the anti-codon that you need to be matching with the mRNA codon to bring a specific amino acid to be added to the inside the ribosome to build up the protein. So it starts with a starting process called the initiation, where it starts, then elongation when they add to each other, and termination when they end. This is the process here. You see a protein made from mRNA inside the ribosome. Here the process just explains translation of mRNA when amino acids have to be added to make a specific protein here. They'll be into vesicle to be released. This vesicle will be released, bringing the specific amino acid. Here to review of peptide formation, we see a DNA making mRNA, having tRNA bring the amino acid to bring the polypeptide together. So that's going to be the end of the translation. We need some specific protein called chaperone that will help making that happen. So this one just explains the different processes, the steps from DNA into protein formation. Cytosol protein by free ribosome. If the proteins to be used in the cytosol, they're going to be made in free ribosome because we have bound ribosome on the rough ER, and free ribosome floating. And after the protein is made, the protein has to be clean. It's called a post-translational modification. Things have to be added to make things more functional. And then some Golgi vesicles become lysosome. Some of them will be different organelles inside the cell. And genes can be turned on and off depending on the situation. And the gene expression, you start with the molecule binding to the receptor, going to the nucleus, right? Expressed in the nucleus, just like a conclusion of this lecture. Look at this one. You have some hormone attaching to a receptor, going to the nucleus, pushing the gene to open, right? The DNA to open so that specific gene can be expressed. mRNA is made, go out of the nucleus to the cytoplasm, onto the ribosomes, pick up the amino acid by tRNA, go into the Golgi, the post office of the cell, and then the vesicle is released. So this is how things are made inside of a cell. We call that gene regulation, like how gene is expressed in the cell, right? So we now understand how things are processed, proteins are made in the cell. But we have other stuff in the cell that are made that are not proteins. For example, lipids, but they're made by enzymes that are coded in the specific gene. So now question, what is the gene? Piece of the DNA that can code for a specific protein. What is genetic code? A system that allows to make specific protein from a specific amino acid. How DNA codes for protein structure? By using triplets, right? Codon and anticodon, tRNA bringing amino acid. How amino acid assemble into protein? By inside the ribosome. What happens to a protein after its amino acid sequence has been synthesized? It has to be reorganized, restructured, right? And how gene can be turned on and off depending on the stimulus on the receptor inside the gene, and how DNA indirectly regulates the synthesis of non-protein molecule. For example, if you make testosterone, it is because the enzymes that make those things are actually coded by the gene, even though themselves are not coded by specific gene. All right. Thank you for watching, and I'll see you in class.